Plan for today, make sure you pause this video whenever you need to make sure that you have enough time to copy down the notes or to ask questions if you have them. Now, I can't guarantee that the sub will know the answers to your questions, but ask each other. Hopefully you guys can puzzle it out. If you still can't puzzle it out, send me an email and I will do my best to reply quickly for you. So today's plan, we're going to take some notes on translation. You have guided note sheets. Our subs or mentors might need to make extra copies. I only left a handful. The rest should be up in the library, but I'm not sure if they were run in time. Okay, then after we're done with notes, you're going to work on your spider goats, and you will finish that spider goat project, finish your annotation assignment, and then work on your study guide, which is a green double-sided page. If you need extra copies, look at the front table. Tomorrow, I will help you get ready for the test in more detail. All right, so yesterday we talked about the 3 types of RNA. Messenger RNA is just that chain of amino acids. tRNAs are the transfer molecules. They carry amino acids to ribosomes. And ribosomal RNA, they build the ribosomes, which will then in turn build proteins. So let's go ahead and start talking about the process of translation. At the top of your note sheet, again, you are going to be copying down the central dogma. Step 1 was DNA making more DNA through replication that occurs in the nucleus. Step 2, we make DNA into RNA. That also occurs in the nucleus. Step 3 is changing that RNA into proteins, and that occurs at the ribosome. So today's step 3 of the central dogma is called translation, making protein. To do step 3, we need to talk about 2 key components of translation. The first one is the mRNA codon. We mentioned yesterday that we read DNA 3 letters at a time, and if that frame gets messed up, it's a mutation. So that section of 3 nucleotides on the RNA, so in this case AUG is a frame or a section of 3 nucleotides, we call that a codon. Now on the other side of things, we have a tRNA anticodon. The codon, anticodon, they have to match up. This is when Gordon Ramsay yells at his little delivery boys and says, hey, you brought me baking soda instead of baking powder, get out of here. So this needs to match. The 2 instructions need to be a match to deliver the correct amino acid. We're going to play this video for you. It's the same animation we've always done. I will narrate for you. This is the RNA. It is leaving the nucleus, that weird purplish looking thing behind with all the holes in it. That is the nucleus. This right here is our mRNA code that we've just copied through transcription. A, U, C, and G. The long message. This blue thing is the ribosome. It comes in 2 pieces, and it grabs on at a place called the start codon. The RNA is pulled through, just like that tape is pulled through cassette tape player or VHS tape is played through a VCR. And as you can see, all those little green molecules are floating around in the cell nearby. The green molecules are tRNA molecules, and they have on them those little red dots. The tRNA is going to deliver amino acids. The red dots are amino acids. And we've got 20 different varieties of amino acids, and they all have a purpose in building protein. We just need to instruct the ribosome on which one needs to be delivered next. So if you cut the ribosome in half and look on the inside, you can see that the mRNA is being pulled through 3 letters at a time, and the matching tRNA comes by, makes a match down here at the bottom. Sorry, this is showing that it's pulling through 3 letters at a time. And the tRNAs are dropping off their amino acids, making sure that they're the correct match. And those amino acids are the red dots being dropped off here at the top. If it's correct, we add it to the chain, and those all get strung together to create a growing protein chain. And this protein could be anything from dietary protein like muscle fiber or things like that, or it could be an enzyme, or it could be something as complicated as hemoglobin. It just depends on what the mRNA, the recipe, calls for. So once again, the tRNAs, these greenish triangles in the background, those are the delivery boys. The amino acids are the red ingredients here. The ribosome is the chef assembling these ingredients into our finished product, which has just been released. All right, so we're going to go through all of those. We talked about the codon up here being this yellow stuff on the RNA, 3 letters at a time. The tRNAs have the anticodon, and those are the green triangles that are in our animation. So phase 1 is initiation. we look for something called a start codon, the start of the recipe. And that is the same sequence in all of biology. We're looking for 3 letters, A, U, G. Every living thing starts the process of translation at that 3-letter sequence. Phase 2 is elongation. We're going to make that protein longer and longer. So we're going to pull the mRNA, this yellow code, through the ribosome like a cassette tape, like I mentioned. The ribosome just focuses on what's the next thing I need, what's the next thing I need, and it just keeps shifting that code on mRNA through 3 letters at a time. The tRNA, this green triangle, is going to drop off the amino acid that is instructed by the mRNA, and we just repeat that process. So this guy dropped off his amino acid, the red dot, and he moved along. This guy is dropping off his amino acid. He's going to get kicked out and move along. This guy is next in line. He'll drop off his amino acid, and the process just continues. You don't have to draw what I drew here, but I did leave you space if you wanted to. So here are my codons. I have 4 codons, or 3-letter sequences. In yellow, the mRNA codon. These are my amino acids that I've already dropped off. Here's my ribosome, focusing on this codon right now. The tRNA is dropping off the correct amino acid. It's going to get added to this chain, and then we'll move along and focus on the next one. Phase 3 of this translation process is to just stop and process that protein. So we're going to look for a stop codon. There are specific codes on the mRNA that say, you're done, this protein is finished. So we release that protein, and as you see in this last image here, it tangles up and goes through a folding and packaging process so that it can then be used by the cell wherever it's needed. So I'm going to go through the process of doing that. You should now have a little half sheet that looks like this, an amino acid codon wheel. It might be on a full page. Cut that in half and share between you and your table partner so that everybody has one of these half sheets. And I'm going to go through the process of how I change this codon, CCA, into the correct amino acid part of the protein. So to do that, video, sorry it's so shaky, I'm going to look for these letters CCA on my codon wheel. So I start in the middle with the biggest letter, C. That leads to C, which leads to A, and that gave me the amino acid proline, so I'm going to fill that in on my diagram, proline. The next one is GGG. So I'm going to start in the middle, G, 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 and that will lead me up here to G for glycine. The next one is CUC, so again I'm going to start in the middle with this C, go to the next smallest pi wedge, U, and then the next one, C. So CUC led me to leucine, which we abbreviate with the letter L. My pen was slipping there. So CUC gives me the letter L for lysine. And then the last one is UUG. Oh, I'm getting dizzy. U, then G, leads me to leucine. And leucine, I'm sorry, these are both different versions of leucine. And that also is abbreviated with the letter L. So if we look at our correct amino acids, we have P for proline, G for glycine, and L for leucine. Lots of different ways to code for leucine. If you look around your wheel, you've got lots of letter L's around the outside, lots of ways to make that part of a protein. If we talk about doing all 3 steps of the DNA process, what we're going to have is DNA making more copies of itself, replication, looks like that. RNA being made, that's the process, DNA to RNA. These letters are going to change to U, A, G, and C, and it looks like this. We're only unzipping a portion of the DNA, making a simple message out of that. And then translation, we're going to use that simple message. We're going to use a ribosome. We're going to connect all these amino acids to make a protein, and that is where we're reading that message 3 letters at a time and plugging in those correct amino acids. So that is the process, all 3 stages, DNA to RNA to protein, the entire central dogma.